Okay, and now we're excited to hear from Kit Curtius, who's an assistant professor at UC San Diego. And she'll be speaking on estimating clonal dynamics using coalescent theory and branching processes.
Thank you, Jasmine. Thank you for the invitation to present. The microphone enough working? Is that okay? Can you hear me in the back? Okay. Oh yeah, that's what I was wondering. I guess they'll shout if they can hear me. Okay, so today the outline for my talk is going to be mainly the first part is going to be about clonal evolution in the hematopoietic system, and a lot of this work and beautiful results that you'll see are spearheaded by my really talented PhD student Brian Johnson in my lab. And so the background is going to include about clonal evolution in the hematopoietic system and myeloproliferative neoplasms, or MPNs in particular, as our cancer type. This work was also done in collaboration with folks in the UCSD math department. So Dr. Jason Swinesberg, who's here with us today, spearheading a lot of the mathematical results, and his PhD student, Yibo Shui. So I'll talk about the model and some results for normal blood and MPN clones. And then if I have time, I'll talk to you a little bit about our current work in solid tissues, so colon and esophagus. But I'll probably take a leaf from Allison's book. And if we don't have enough time, that's okay. We can focus on one story, but we'll see how far we get. Okay, so I want to start with one slide with kind of my perspective with the lab and what we focus on. And so one of our big goals is trying to improve cancer control by bridging different data types. And one thing that I noticed very early on in my cancer career is that current cancer screening guidelines for high-risk populations to catch early cancers, which often have a better treatment prognosis, is really based on very limited data and mostly on expert opinion. So ideas of when to start screening patients who have a pre-cancerous condition called inflammatory bowel disease, which is a big disease we study in the group, is done in this way, where once you're diagnosed, 8 years later, you're suggested to have your first colonoscopy to look for early neoplastic lesions. And then based on the findings from that, you are suggested to come in every 1 to 3 years based on clinical risk factors. The decisions on these years and intervals is basically just looking at epidemiological and clinical data and making some sort of best guess of when you should come back. So, mostly expert opinion, not usually derived from much more data than that. On the other flip end of the spectrum, we know that the underlying reason why cancers develop at certain timeframes is that they're all driven by cellular dynamics. I know I'm speaking to an audience that knows this well, but two of the main precancers that we study in the lab are Barrett's esophagus and IBD, as I mentioned. And we know from various studies that clones that evolve in these tissues and accumulate more mutations actually are what's driving cancer evolution and cancer progression. And so even just in Barrett's esophagus, if you just count the number of genetic clones, it shows that that is a good risk stratification tool for future esophageal adenocarcinoma in these patients. In the same inflammatory bowel disease, large fields form of clonal populations that just give more fuel to the fire of potential driver mutations for colorectal cancer. And so kind of my perspective is that computational models are really needed to answer some of these complex questions that bridge multiple scales. So we're talking about mutations that happen at a single cell level, all the way up to how many cancer cases are we seeing in the population. And so complex questions about costs and benefits that we've heard about a little bit this week about treatment, and incorporating these different data types. I believe that we can't do these on the back of an envelope. We need models to help us answer these questions. The kind of term that I've given this is sort of quantitative cancer control, and this can help us answer clinical questions like when should we recommend screening, how should we use biomarkers in the process, and how much will all these efforts cost. So focusing on the blood now. So this is a basic diagram of how the blood system is formed and evolves over time in the human body. So during embryogenesis, the hematopoietic stem cells are formed. And we think that in adulthood, there's a somewhat constant number of the stem cell pool, which we'll call HSCs, of somewhere probably between 50 to 200,000. And during this process of aging, inconsequential mutations can occur in this HSC pool, but occasionally a certain mutation might happen that confers a selective advantage in the hematopoietic system. And this clone will become overrepresented in the population of stem cells. Such a mutation that's common is DNMT3A, which is an epigenetic regulator. And this process has been termed clonal hematopoiesis. So these clones outgrowing during aging process, and it's very common. So we think over 20% of people over age 70 have these clonal outgrowths. And unfortunately, they've also been associated with poor outcomes. So things like cardiovascular disease, death of any causes seem to be increased in these patients who have these clonal hematopoietic stem cell expansions. Also, you're at higher increase of developing cancers. So an additional driver mutation might start what would be a precursor to a secondary acute myeloid leukemia. So secondary AML are formed oftentimes starting from these JAK2 V617F mutations, and they're often more lethal than even an already lethal primary AML, and basically have this clonal process where additional mutations confer more and more selective advantage, and you can see these in sequencing. Now, I've totally glossed over the fact that HSCs are the top of this hierarchical structure of the blood system in the bone marrow and the peripheral blood. And so they are the progenitors of multiple types of cell types, and we'll talk a little bit about that. But the main point being that HSCs are the long-lived stem cell population, that mutations in these will be in the progenies of all these cell types, and we kind of gloss over the fact that there is this complex structure and focus on these stem cells. And one of the basic questions that me and Jason and Brian and Yubo asked early on is, how fast do these clones expand, and does that have any clinical relevance? And so Allison just gave a wonderful introduction about phylogenetic trees, which is really useful for this talk, talking about the kind of data that we ended up applying these models to, which I wanted to introduce first. And so what we used was basically single cell sequencing, but how they did this in this Nature paper in 2022 from the Sanger Institute is they had normal individuals without cancer, and they sampled cells from either the bone marrow or cord blood in infants or peripheral blood in adults. And then they single cell sort them and put them in a dish and expand them into a colony. The nice thing about this process is you get this colony form that was from a single cell. So they have all the mutations from that first cell, but it gives you more genetic material to sequence. So then you're able to do whole genome sequencing of this colony. And what you get is a representation of that single cell. And you can build these phylogenetic reconstructions that Allison introduced, where each tip can be considered a single cell. Of course, you get mutations, and she talked about mutation trees. And these can be converted into what we say maybe in phylogenetics is ultrametric tree, which is time-based trees. And these are the kind of trees that you see. So the age of the patients is shown at the top, so 76, 77, 81. And what you can see is that these clones do form in these patients. So sometimes there's a clone with a large clade that has some sort of known driver mutation in the trunk. And so we understand why that maybe had a selective advantage. And a lot of times there's clones that seem to have no known driver when we look at the sequencing data. So this is just showing kind of that these clonal expansions occur. And these would be the kinds of clones that we are curious about how fast they're expanding in the human body. Then the same group published another paper in Nature looking at cancer patients. So doing the same type of single clone population outgrowth and sequencing, the trees look something like this. And what you can see is that oftentimes there's really large clonal expansions that occur. And that's probably because these certain mutations confer this very high selective advantage in the hematopoietic system. And so maybe just like focusing in on this patient 6646, you see that they have an initial DNMT3A mutation that occurred somewhere around age 20, which divided and produced different clades that had subsequent driver mutations in JAK2 and CBL that also clonally expanded. So just a little background about myeloproliferative neoplasms, or MPNs. These are rare blood cancers that happen when bone marrow makes too many blood cells. So the three most common types of MPN are polycythemia vera, essential thrombocythemia, and myelofibrosis. And the symptoms come on slowly, and then they sort of worsen as the condition progresses, as opposed to later stage AMLs, for example. This gives you the opportunity to sample from these patients over time, because sometimes they're not even on treatment for quite a while, and just watch and see if symptoms progress. And it can lead to poor outcomes, so heart attack, stroke, and like I mentioned, these AMLs. And so again, focusing in on this one patient, we have samples then from these clades. And oopsies. So this, for example, from this JAK2 clone, we have 51 cells that were sequenced. From the CBL clone, we have 13. And from this expanded clade with no known driver, we have 18. So this is the kind of clonal populations that we were focusing on for the modeling work. Okay, so let's talk about the model development. And so mathematical models of clone growth in the hematopoietic system have been coming out throughout the years and various different types. And what a lot of these models listed here at the bottom have in common is that they have this early exponential growth phase. So an exponential model, obviously a logistic growth model, right? Fisher with selection also has exponential growth. And so what we started out with is this idea that we can just describe this process of exponential growth at the beginning of the expansion as having a symmetric division rate, as Alison mentioned with rate lambda, an asymmetric division rate that has a differentiated cell and does not increase the stem cell population, and then the cell differentiation or death rate mu. And then this expansion process will be then described by this growth rate, lambda minus mu, or R. And then mutations then accumulate in the clone and cause these branches to have a certain length based on the number of mutations post initiation of the clone. Now, something I want to kind of emphasize here is that as we talked about in the previous talk, if you were able to sample every cell in the clone, you get something on the left here that we could call a population tree, where you know every coalescence time for all the cells in the clone and all the reconstruction. This is obviously infeasible. We're never going to sample every cell. So what we get is a sampled tree. So if we were just to sample 10 cells, luckily from this clone, we would get a phylogenetic reconstruction that looks like the right-hand side. And that has a very different looking structure, obviously. And the important thing to note is that the main idea of why the math works here is that for sample size n, little n, that's much smaller than the total number of the clone in the hematopoietic system, then coalescence times that you have in your tree will be all occurring near the birth of the clone. So they're all kind of bunched up at this initial time in the initial years of the clone initiation, whether you have later exponential growth, logistic growth, even competition later, you're still going to capture those early growth dynamics just by looking at these early coalescence times. And sort of in classic coalescent theory, these are sort of called star-shaped trees, where you have these long external branches. That's something that I'll say again and again, is internal branches in red are the ones that are shared among different cells, and the external branches in blue are the mutations that are private to a single cell. Okay, I'm only having one slide on the math of this because a small part of Jason's talk on Friday is about the whole intuition behind this, and it's really beautiful. So definitely stay tuned for that. But I do have to tell you a little bit about it. So as Alison mentioned, a lot of phylogenetic inference tools looking at growth rates have been focused on Bayesian inference. And some of the examples of methods are Markov chain Monte Carlo, integrated Laplace approximations, approximate Bayesian computation is popular. And these are some examples of some tools. So BEAST that she mentioned, Phylogen, and PhyloFit, which we'll talk a little bit more about. And she already explained how you have these empirical trees and you try to sample from these posterior distributions of some model parameters eta given some tree and some model structure. You can simulate this with master equations as they do in BEAST and try to fit these summary stats to find the tree that most fits the data. This can be a difficult process if you have a very complicated tree, et cetera, but most tools have been focused on this. What we did was just looked at actually the mathematical results of how these coalescent processes actually form in a tree and derived analytical methods for the growth rates and the confidence intervals. So I'm just going to give you the result, and then again, Jason will explain more about the math, but the basic really nice result that we were able to take advantage of was from Amaury Lambert, published in 2018, which is writing down actually some equations for birth-death process trees where you have the known number of sampled tips. This is different than some other math that you might be aware of sort of birth-death sampling processes from Tanya Stadler and others. This is actually, if you know the number of sampled tips, then we can actually write down using something called a coalescent point process of what the distribution of these coalescent times are by drawing these vertical and horizontal connections to these vertical lines. If you take this result from Amaury Lambert, what Jason and you were able to do was when you let t and n go to infinity, then you actually get a very simple equation for what the coalescent times are. They're these h's, where these u's are logistic random variables. This w is an exponential random variable, and you're able to write down what the growth rate is in terms of these simple equations. And that allowed us to make two methods, one that uses max likelihood to derive what this R is. And then amazingly, they're even able to write down analytical confidence intervals, which was a long tedious process for them, but so amazing because that's usually a thing that takes lots of simulation to obtain. And then another method that we call lengths that has another kind of intuitive understanding where you look at the number of tips sampled divided by the total number of the internal lengths, and that gives you an idea of the growth rate as well with confidence intervals. Okay, that's basically most of the math that I'm going to explain on that. So I'm going to jump into simulated data. Another thing about having that Amori-Lambert paper is that we have this really easy way to simulate trees now, which used to be kind of difficult. And one thing that we put into our R package that has all of this kind of methods is a way to just create random birth-death trees very easily. And so we're able to simulate data very easily. That's the point. And what we looked at in comparison to our methods, which we call lengths and maximum likelihood, is this other method called phylo-fit, which basically depends on the Kingman's coalescent with a logistic growth function, deterministic logistic growth function. And then we wrote a birth-death Markov chain Monte Carlo, which again has this birth-death sampling process that's similar to what's used in BEAST, and compared how they work. So one thing that we varied was number of samples, which Allison talked about too, as in, how can you get a reliable growth rate estimate based on how many things that you sample? And I really like this kind of, marrying of math and experimentalists, because of course, without any prior knowledge about how this process works, you would just be like, oh, well, I don't know, maybe 100 or something, but actually being able to say how many you really need, I think is important and saves us a lot of time and money. So what we found is that for the true growth rate estimate, where we know the ground truth, somewhere over 30 cell samples seems to be sufficient to get back to the growth rate. And we did lots of simulation. Of course, the more cells you sample, the more understanding you have of that early coalescent structure. And just one thing to know, just because we're mathematicians and it's interesting, is that what we noticed, and was noted by Tanya Stadler and others in the past, was that if you use this Kingman's coalescent with this deterministic approximation, you don't correctly recover the confidence intervals. So your confidence intervals are too narrow and too overconfident. So to properly get the right confidence intervals, you need to actually understand the stochasticity of those early birth events. And that's something that can sort of be mathematically shown, that you're going to be underestimating the 95% confidence intervals if you assume this deterministic growth function only. And I guess the biggest plus of our method, which I mentioned, is that it's basically instantaneous. We just have an analytical result, and then all these others, take simulated time. We also looked at what happens when you vary R. So how big does the growth rate need to be to actually get this to work? Those should be R's, these boxes. So if the growth rate is too small, and you sample not very long after the clone is born, then you're not going to have enough of these early coalescence times to properly capture. But then once you have a sort of, reasonable growth rate, then the model seems to capture the growth rate very well. And future work with Jason that we've been talking about is how can you look at a tree and have some sort of metrics to understand if that tree reflects the growth dynamics that you are anticipating with your model. And there's probably a few different quantities to look at besides whether the coalescent times are logistically distributed, et cetera. But for the very basic purposes of whether R is big enough for your method, Brian came up with this diagnostic that basically the sum of the external branches divided by the sum of the internal branches should be greater than or equal to 3. And basically that just means that the tree is sufficiently star-shaped to use this. And what was nice is in the real data, we find that it's always greater than 3. So it seems to be, all these trees are appropriate for this type of model. Okay, now I'll show you the results that we published and some unpublished things. So this was the sequencing data that came out. It was actually really nice because we started working on this project in 2021. We were making all this crazy math and stuff, and we were like, what do we do now? And then all these papers were published that had all the data that we needed. So it was really perfect example of things happening at the right time. So these papers were published in Nature. Some of them had myeloproliferative neoplasm samples from patients. Some of them had normal clones, so just regular clonal hematopoiesis from patients. And then this previous paper also had MPN clones. And for a few of the clones, we had longitudinal data, which I'll talk about single cell data. And one thing that we wanted to check was, are these clones evolving neutrally after they're initiated? And a lot of you will be aware of work by Rick Durrett and others that derives this equation for if you have neutrally evolving process and mutations accumulating in a clone, then you would expect that the number of mutations inherited by K of the N sampled individuals goes something like 1 over k times k minus 1. And so when we wrote down the frequency of the mutations in the descendant cells, we found that the theory matches perfectly with the data. So it seems to be that these clones that are well mixed in the hematopoietic system are evolving neutrally, or like our designation of a clone. And sometimes we would remove a subclone if it was very obvious, actually evolved neutrally. And this is a teaser for Jason's talk again on Friday, because he derives this math for the first time for a spatially structured model, and it's really nice. So, but it was really nice to see that, okay, there's probably no extra selective advantage happening, and these things are evolving neutrally after initiation. So these are the results from those studies. So we applied the model to normal clones and MPN clones, as I mentioned, and we've also compared it with this method phylophit in the normal cells. And what you're seeing is each column is a patient, and then the 3 methods are each of the bar plots there. The number of cells sampled is the N, and you can right away tell that there's very concordance between phylophit and our methods. And that the normal clones on the y-axis have a smaller growth rate in general than the cancer clones. The colors are the different mutations. And one thing that I mentioned before was that we had, some patients had multiple time points of single cell data. And so what we wanted to see is if you can just take a single time point, can you recover this growth rate no matter what time you sample? And so what we did find was that for these few patients that we had, there was very close concordance in their growth rate estimates, meaning that we are capturing this early expansion phase no matter, that's not affected by time of sampling in general. We compared this to some data that was out there for other cancer types out of interest. So chronic lymphocytic leukemia, or CLL. This was published in 2019. Some estimation of this looking at not clone-specific growth rates, but just the white blood cell counts as like the total tumor burden growth rates. And we found actually that these were very closely in line, I think between 0.1 difference of mean growth rate here between the MPN clones and the CLL clones. So that was somewhat interesting that these clonal populations are in line with at least the total tumor burden in CLL, which again are lower than the max likelihood, which is more like 0.2 growth rate. And yeah, just to put that again in another plot, the MPNs, of course, were higher growth rates than the normal clonal expansion seen. Even though you could see a huge clonal hematopoiesis clone in a normal patient, you really can't just look at that tree and just determine if it has a high growth rate or not. We were able to quantify that. Another thing we were able to quantify that is probably most people believe, but rarely quantified, I think, is that people that had 2 or more drivers in their clone had higher growth rates than the ones with 0 or 1. So there seems to be some sort of additive effect of having more mutations, you grow faster. And then one of the exciting things is if you look at the patients who have MPN, and we know what time their clone was initiated from those phylogenetic trees, and you split up the cohort between those that had lower than the mean growth rate and higher than the mean growth rate, and then looked at when their symptoms onset and they were diagnosed, there was a significant difference between those 2 groups of patients, where the ones with fast initiating clone expanding progressed to symptomatic cancer faster than the ones that had slow expanding cells. So that was sort of interesting that there seems to be some sort of clinical relevance to these types of evolutionary measures. And just to sort of drive that message home is then we just looked at another kind of feature or metric that you can look at in this kind of data, which is just what's the biggest clone? So what is just the aberrant cell fraction of the biggest clone? That was actually used in some of the initial Nature papers, and we kind of made some points in our papers, and now they're not using that as much anymore, because basically you don't see any difference between the patients just by looking at what has the largest clone. And it makes sense, right? Because it's probably only telling you that the clone is big enough to be symptomatically detected, and it doesn't really tell you anything about the past evolutionary history. So just more reasons why taking sort of an evolutionary perspective about this is probably more meaningful than just looking at things like burden of the clone. Okay, then we had some longitudinal data on the same patients and the same clones from additional data from these groups shown at the bottom. And so what we had was vast distributions of the same clonal populations over time. And so we just fit logistic growth functions to this data and compared with our single cell time point data. And so what we found is that in these 3 patients that we had the same clonal information about, we saw consistent growth rates with the longitudinal data. So that was somewhat comforting to see that this is growing in a way that we expect, and also that you can get growth rate dynamics from single time points was nice validation. And then also our question was, is this repeatable among patients? Is there something about the certain mutation type that also is reflected in the growth rate. And we saw, variable, but still somewhat consistent growth rate across different mutation types. So, maybe between patients, there's some similarity. If you both have a JAK2 mutation, it grows at a particular rate, and that sort of repeatable evolution in different patients. Yeah, please.
So this logistic growth assume you know time zero.
Sorry?
Logistic growth, you need some kind of time zero. You set that to your when the mutation happens, when you want to how you get when you go back one slide.
Yeah. So
what is time? How do you know time zero when you do this?
So we just know the person age. So we have time points. And yeah.
Time equals zero, the person age.
Yes.
The
Yes.
occurrence of the mutation.
It's the person age. Yeah. Yeah. For the longitudinal data, we pretend we don't know about.
The mutation happens very early. Sometimes it may happen at year 30.
Yes. Yeah. So we don't, from the single cell trees, we know like when the clones started around, but for the longitudinal data, we don't know. So, it's going to happen sometime back here. I guess I should have put that on there maybe to show that the single cell trees are before that time. That help? One of the last kind of things that we put in the paper was about, clones in the same patient. So back to our patient 6646, we have single cell data on the JAK2 clone, the CBL clone. We saw from the estimates that the JAK2 clone was growing faster than the CBL clone. And then we had, again, bulk sampling from these clones from the same patient. And we saw that, the JAK2 clone was growing and the CBL clone was decreasing. So this is just anecdotal, it's just a single patient, but, maybe some idea that these growth rates are also signifying potential outcomposition of one clone versus another clone. What else do I want to say about this? I will just reiterate that the nice thing about the method is if you want to learn about the early growth rate, you're not having to deal with issues of treatment effects or anything because we don't care what happens in the late stages of the tree. We only care about those initial times near the birth of the clone, which aren't affected by treatment yet. So this is an idea we had after publishing this work was, can we also use this model to sort of understand bounds for the total numbers of stem cells? And Brian had this good idea of simulating the process by looking at different values for the effective N of the total stem cell pool size. And so what effective is basically N tau, which is the clone size times the, I should say total population size there, and times the time between symmetric division. So this is something that you can't sort of identify separately. You have to look at them together. And so when you simulate these trees, we know the times of the initiations, we know the growth rates, for example, and you have different N effectives. What we saw is that when you compare the max likelihood estimate to the phylophit estimate for starting at, say, 10,000 stem cells, you don't see good concordance between the methods. And the reason that is that there's not enough stem cells for the process to have that structure of star-shaped trees. And when you increase the N effective size, you have less likelihood of having these late merging events where you'll have random coalescence near the bottom of the tree. And then at some point around 60,000, you see very high concordance between phylophit and max likelihood and continued good concordance. When we looked at the real data, you also see really good concordance for those with greater than 30 in their sampled tips. And so what that leads us to believe is that what we're seeing in the real data is more reflective of things that have over 60,000 in stem cell years. And we think for really fast growing clones, there's potential of getting an upper bound estimate on this as well. And so that's data that we're reading on now. But yeah, this total number of stem cells has been kind of an elusive number that people try to estimate with models. So we wanted to see if it was in line with what other people were finding. One other thing that we're interested in that's sort of very much a work in progress, I guess, is what about things that happen in utero? So we know that clonal mosaicism from mutations that happen in utero can be very influential on cancer risk in the future and including childhood cancers. And so we were kind of interested in looking at the trees that of the non-expanded clones. So just looking at the tree itself and everything at the bottom. So here's an example of a patient have their mutation tree at the top, their ultrametric tree at the top, at the bottom. And if you zoom in to say the first 55 mutations that happen, that's what they think is about the average number of mutations you have in your hematopoietic system when you're born. Then the tree will look something like this. And again, whether this tree is following this perfect amount of growth dynamics that we expect is an open question, but let's assume that it is. And then we also had cord blood samples, like I mentioned. So these are taken from immediately after birth, and we can also do the same thing by scaling them to time of birth. And so the question is, if you apply the methods to these type of trees, what do you get in terms of the early expansion rate of the hematopoietic system? And so this is what we found using this type of data across the studies where we had such information. We had really high N numbers because these are the cells that aren't in the subclones. And we saw pretty good consistency across the different patients, even though some are from cord blood and some are from adults. So trying to just see if you can actually estimate embryonic expansion rates from different time points seemed to give consistent results. And something that I just did the past couple of days was I was wondering, assuming that you start from, say, a single stem cell, or we're not quite sure, how many stem cells lead to all lineages of the hematopoietic system, and you use, say, the mean growth rate here of about 23, which is extremely high per year, what does the curve look like? And so just a very preliminary result is you write down this number. Also, I have 36/52 here weeks because we think that the hematopoietic system starts forming around 4 weeks post-conception. And so when you use just n not equals 1, so just assuming that the whole system starts with 1 cell, you get this picture where you have this logistic growth function that levels off around 40 weeks, which was not expected. But if you assume that n naught is much higher, say that maybe it starts with 50 stem cells or whatever, this curve is quite shifted. So this was kind of nice to see that this is maybe a reasonable type of expansion rate to expect in utero. But of course, it's a very complicated process happening during that time, but sort of some cool preliminary results. So I'm thinking about pausing. I have some kind of ideas of what we do with multi-type processes. Maybe I'll just tell one story and say the rest. But basically, what we've been interested in is these multi-stage processes. So not just a single subclone, but what about the clones in between? And the reason we've been interested in this is because we're interested in pre-cancer development, field cancerization that eventually form cancers and have multiple mutations that cause clonal expansions. And so it's been mentioned this week that multi-type branching processes are a way to model this process. And they tell us about certain stages of the different types. And one thing that we have been studying is GI cancers, which have oftentimes multiple stages. So colorectal cancer is one that has these early initiation events for adenomas with biallelic inactivation of APC that can cause independent expansions of adenomas. These can sojourn for many decades before another mutation might cause initiation of a colorectal cancer. When you write down a branching process model for this, it looks something like the bottom with different mutation rates and growth rates, like we've discussed. And what was some of the earliest math models applied to these were looking at population data. So looking at incidence data and mortality data, do these curves tell us something about this process of multiple hits before we even knew anything about genome sequencing or anything? So early papers, so Armitage and Dahl and Mughal-Parvin-Benzin looked at these incidence curves. And when you write down what the epidemiological hazard function is for these models, actually you get something that looks a lot like this curve. So there's this early exponential growth phase, a slope, and some sort of intersection with age. And what you do is use this kind of registry data, say for colorectal cancer in the US, and fit the model to these hazard curves. And parts of the hazard curves tell you different parts of the model. So the pre-malignant adenoma phase is captured in the early expansion phase when you write out the analytical result for hazard function. The sojourn time of the tumor is captured in this intersection. The slope tells you something about the early initiation events of the adenomas. And so we've been applying these models in various settings, and the goal is if you have these parameters, then you can make predictions about what happens in the future. So we've done this in various cancer types, and one of our main goals is looking at this in esophageal cancer. And for this one, we have a special initiation event to Barrett's esophagus, like I mentioned before this evolutionary process can start. And so we fit these models to these incidence curves differently for men and women in these multi-stage way, and are able to, capture some of these parameters. We look at men and women separately because they have different incidence rates. And one of the nice results that clinical data wasn't able to tell us was that the model predicts that there was about 10,000 EACs in the US in 2010. And we use life tables and multiply by the hazard function to get this cumulative hazard. And the nice thing about that is that that marries up with how many cases we expect from registry data. And so it was able to answer this long held question of, do all esophageal adenocarcinomas come from Barrett's? Because we actually didn't know if that was the case. And that's been now like corroborated later in some molecular and epi work. So that was a nice case of modeling telling us an answer we didn't know. And basically we've been using these models now to focus on Barrett screening and when we should screen people for Barrett's. I included these slides because I thought it would be interesting to people that we write down kind of an optimal control problem for this, trying to find windows of opportunity of screening during this process by looking at probabilities of screening times between early precursors and cancer development. You can write down objective functions to try to maximize to find optimal timing windows of these screening times. And I guess maybe the last thing I'll show is this, which is we found optimal times for screening for Barrett's, and using trial data from 2020 in the UK. These were validated this year, and right now Barrett's patients are screened for, or GERD patients are screened for Barrett's at age 50, but we were suggesting that they should be screened later, actually, to capture more and have very low cancer risk. And so now the newest trial in the UK, that's randomized controlled trial for 120,000 people, are indeed initializing screening at later ages. And so I think this was a really nice example of how modeling can inform clinical trial design and future efforts in terms of public policy for better ideas of when to do these types of interventions and public health programs. I think I'll end there. So we have new studies looking at spatiotemporal evolution in Barrett's that are funded by the NIH. If you're interested in more about this, please get in touch. We're trying to measure various dynamics that happen in the Barrett segment and see if these help inform our models, with the goal of sort of personalizing surveillance by looking forward in time and simulating an esophagus with multiple clones progressing. And someone had mentioned digital twins this week. And we have something coming out that talks about this in terms of using patient-specific measurements to update over time and make future predictions about treatment decisions. Okay, with that, I'm gonna skip this part and summarize. So we derive these analytical estimates and their confidence intervals, so it eliminates the need for Bayesian inference for this problem, this specific problem. The growth rates were higher in the multi-hit clones, and they progress symptomatic cancer faster. And this is this measure of evolution that may predict clinical outcome and might be used to, triage patients with MPNs in terms of whether they should have more aggressive treatments and interventions. Brian made this really nice R package called Clone Rate that's able to simulate these trees and also do all this estimation. Really easy to use. Encourage you if you're interested and you have phylogenetic trees that you'd like to apply to. And then our future work and current work is using these multi-type branching processes in GI cancers. So thanks to my group, especially Brian for leading so much of this work. Thanks to Jason and Yubo for this wonderful collaboration we started and are continuing. I just put my contact information up, especially for people in the online group if they want to contact me. And happy to take some questions. We've been really grateful for lots of funding the past year for various studies, and excited to hear you guys' feedback and questions. So thank you.